Welcome to Prime Immunology. This is the first of 3 videos meant to provide you with an introductory preview or review of key concepts in immunology before your block begins this fall. The 3 main arms of the immune system include the innate, adaptive, and complement systems. Though these 3 branches do interweave within the intricate web of immunology, we will learn about the basics of each separately. Throughout this module, we will address key concepts of innate immunology. The innate immune system, as its name suggests, is made up of physical barriers, cells, and proteins that are always present and ready to fight off infection. Working from the outside in, epithelial cells of the skin, respiratory tract, and GI tract play a crucial role in keeping microbes outside of our body. Tight junctions between these cells provide a physical barrier to entry, mucosal and ciliary movements help trap foreign bodies, and airflow through coughing and sneezing expels them. Chemical barriers also exist to inhibit microbial growth. Acids in your stomach and enzymes in your tears, for example, can neutralize or even destroy pathogens. And our body is filled with its own population of commensal microbes that play their own role in fending off unwanted ones. For the microbes that do pass through these barriers, innate immune cells play a role in mitigating damage. The 7 cells we will touch on today are neutrophils, basophils, eosinophils, monocytes, macrophages, dendritic cells, and natural killer cells. Granulocytes are cells that are characterized by internal granules that contain enzymes or other active substances. Neutrophils, also known as polymorphonuclear leukocytes or PMNs, are a crucial phagocytic cell in early inflammation. Phagocyte, from the Greek phagein or eating, and cyte for cell, means exactly that. A cell that is capable of ingesting particles. Here, our ingested particles are the invading pathogens. A neutrophil will not only internalize, but also degrade antigen via highly toxic reactive oxygen and nitrogen species. Through this so-called oxidative burst, the invading pathogen is killed. Following a bacterial infection especially, one's blood neutrophil count rises rapidly and dramatically to fight off disease. Basophils are cells whose granules contain histamine and heparin. Though they are physically the largest type of granulocyte, they are also the rarest, accounting for less than 1% of circulating white blood cells. Basophils are highly specialized in that they are responsible for immediate type hypersensitivity reactions, that is, allergies. Lastly, eosinophils contain powerful enzymes that play a critical role in killing parasites. In addition to parasitic infections, these cells are also associated with chronic allergies. Unlike basophils, which stain blue with the widely used H&E stain, eosinophils are typically histologically identified by their pink appearance. Next up are monocytes and macrophages. These 2 cells are presented together because monocytes, which are found in circulation, differentiate into macrophages when they migrate to tissues. Macrophages are then further given specialized name depending on the tissue type in which they are found. Like neutrophils, macrophages are phagocytes. Unlike neutrophils, however, macrophages also act as antigen-presenting cells, meaning that they are capable of degrading foreign peptides and displaying them on their own cell surface. These so-called major histocompatibility antigens can then be found and identified by other immune cells. We will talk more about major histocompatibility antigens and complexes in the adaptive immunity module. Dendritic cells are a very special type of phagocytic cell. Just like macrophages, they also present antigen. In fact, they are the key antigen-presenting cells of the immune system. In your immunology block, you will learn about 2 major types of dendritic cells, one whose main function is to phagocytose antigen for presentation throughout the body, and the other type that is responsible for amplifying immune responses in lymphoid organs. Natural killer cells are a unique type of innate immune cell that destroy human cells rather than microbes. These cells are able to survey the surface of human cells to determine whether or not they are infected or malignant. If so, the cell initiates a cytotoxic response via release of granzymes and perforin to destroy the affected cells and prevent spread of disease. As we have seen, the innate immune system is made up of cells and proteins that are always present and ready to fight off infection. However, how does it know when to fight? The key theme in this regard is recognition. That is, there exists a series of molecules that recognize microbial components. A specific type of recognition receptor that we'll touch on today is the toll-like receptor, or TLR. There are several different types of TLRs, each of which recognizes a specific type of foreign material, or pathogen-associated molecular pattern, PAMP. Examples of PAMPs include LPS, found in gram-negative bacteria, flagellin, found in motile bacteria, double-stranded or single-stranded RNA, such as can be found in viruses, and more. When TLRs, either alone or dimerized to each other, recognize the presence of a microbe, they initiate a signaling cascade that ultimately results in immune activation. You will learn a lot more details in your upcoming immunology block. Before we finish, I'd like to introduce the topic of cytokines, which you will come into contact with often throughout your immunology course and beyond. Cytokines are soluble molecules that act as messengers both within the immune system and between the immune system and other systems of the body. Cytokines can be inflammatory, that is, they upregulate immune processes, or anti-inflammatory. Here on this slide, I've listed a few examples of key cytokines found within each class. This is a summary slide for you all to review what we talked about in today's module. Thank you.